In microbiology, we have this really interesting challenge, right? How do we possibly grow and study a world of organisms that are completely invisible to the naked eye? Well, the secret is all about creating the perfect environment for them, and that's exactly what we're diving into today.
Okay, let's get right to it. We're going to start with the basics of what culture media even is. Then we'll break down the big differences between liquid, solid, and some cool specialty types. And we'll wrap it all up with a super handy cheat sheet. By the end of this, you'll have this stuff down cold. So first up, what exactly is culture media? The easiest way to think about it is like the kitchen of the microbiology lab. It's basically a nutrient-packed recipe. Sometimes it looks like a soup, sometimes like a jelly, that lets us grow microorganisms like bacteria outside of their natural habitat so we can actually see them and study them. Now, you can classify media in a whole bunch of different ways, but for this quick revision, we're going to focus on its physical state or its consistency. It really all boils down to one simple question. Does it have agar in it? That one ingredient is pretty much the dividing line between all the media types we're about to cover. Okay, so let's start with liquid media. Think of it this way. You grab this tool when you have a very specific problem to solve. You've got a tiny amount of bacteria, but what you need is a huge amount, and you need it fast. You need to amplify what you've got. Liquid media is also just called broth, and its most defining feature is actually what's missing. There's no solidifying agent like agar. And because it's liquid, when bacteria grow, they don't form little spots. Nope, they just spread out all through the liquid, which is what makes it look cloudy, or what we call turbid. And you can see the difference right here. The liquid broth lets the bacteria grow everywhere inside the tube, not just on a single surface. This is exactly why it's so good for growing a population quickly. The bacteria are surrounded by nutrients from every direction. So here's the bottom line. You use a liquid broth when your main goal is quantity. Whether you're trying to ramp up the number of bacteria for a big experiment, or you're just trying to maintain a pure stock culture you already have, broth is your go-to tool for amplification. All right, let's switch gears and pick up the next tool in our kit. This one solves a totally different problem. What if you don't have a pure culture? What if you've got a messy mix of all kinds of different bacteria, and you need to separate them out so you can study them one by one? Well, that is where solid media comes in. Solid media is really just broth with a secret ingredient, agar. Agar is this amazing complex polysaccharide that comes from red algae, and it's perfect for the job because it creates this firm, gel-like surface that bacteria can't digest. They just grow right on top of it, which makes it the perfect stage for us to observe them. And solid media isn't just one thing, you know? As you can see here, it actually comes in three main flavors. We've got plates, slants, and deeps. And each of these different formats has a really unique job in the lab. The agar plate. This is probably what you picture when you think of a microbiology lab, right? That big, flat surface is perfect for streaking out a mixed sample. As each individual bacterium divides and multiplies, it forms a visible little pile called a colony. And that's how we get a pure culture from a mixed-up sample. Now let's check out the test tube versions. The agar slant is just what it sounds like. It's cooled at an angle to create a bigger surface area for storing and maintaining pure cultures. The agar deep, on the other hand, is solidified standing straight up, creating this column of media that serves a totally different and really clever purpose. And this is where the agar deep really shines. It's such a simple but honestly brilliant diagnostic tool. See, oxygen can only really seep into the very top layer of the agar. So wherever the bacteria decide to grow inside that tube tells you everything you need to know about their relationship with oxygen. It's a beautiful little experiment. Okay, so we've got our basic liquid and solid media down. But now we get to the really specialized tools. Beyond the basics, there are two specialty consistencies that are used for very specific tasks: semi-solid and biphasic media. Let's put them side by side. Semi-solid media, it has just a tiny little bit of agar, making it feel like a soft jelly. Its whole purpose is to see if bacteria can move around on their own. Then you've got biphasic media, which, just like the name says, has both a solid and a liquid part in the same bottle. It's used for really specialized things like blood cultures. So what happens when you cut way back on the agar? You get this soft, kind of jelly-like texture. It's just firm enough that if bacteria can't move, if they're non-modal, they'll only grow right where you stab them into the media. But if they are modal, they can literally swim right through the jelly, and you'll see this hazy cloud of growth spreading out. And the design of biphasic media is just
It's so clever, especially for something sensitive like a blood culture. The sample grows in the liquid broth part first. Then, when you want to create a subculture on the solid part, you don't have to open anything up. You just tilt the bottle, wash the broth over the built-in agar slant, and you're done. It massively reduces the risk of contamination.
Okay, we've covered a ton of ground. So this is the moment we've been building up to. Let's pull everything we've talked about together onto one slide, the ultimate summary of media consistency. Here it is, your cheat sheet. Let's run through it. Liquid, no agar. It's all about growing large quantities. Solid, that's your 1 to 3% agar, making a firm gel perfect for isolating colonies. Semi-solid, barely any agar, giving you that soft gel for motility tests. And finally, biphasic, which combines both liquid and solid for those specialized systems like blood cultures. This table is your key.
And this is so much more than just memorizing facts for an exam. Choosing the right media is absolutely fundamental. It's the first decision you make, and it dictates what you can grow, what you can see, and ultimately what you can discover. Getting this first step right is everything.
So don't just think of these different media as lab supplies. Think of them as a toolkit. Each one is a specific tool designed to answer a different question or solve a different kind of problem. Now that you know what the tools are for, the only question left is, which one are you going to use to make your next big discovery?